we will schedule the assessment on the last day of the course. But this time, as some participants requested some 2 to 3 days gap to prepare for the final exams, final assessment, we are planning to conduct the final assessment on 26th January. That is, on 26th January, people, we all of us will be on leave, so that we will be free on our home or residence. So it will be easy for all of us to attend the test on the day. So we are planning to conduct the final assessment on 26th January 2024 at 6 p.m. to 7 p.m. Okay. So on the day 26th, there is no necessary for to join the session through Google Meet. We'll be sending the link to you. We'll be sending the link of the Google Forms to you. It contains around 20 questions. So participants should attend all the 20 questions. And please note that this assessment is not to give you marks or give you grades like that. So it is, the purpose of the assessment is to measure the efficiency of our course, and it is a formality. So I request all the participants to attend the course. There is no passing minimum for this assessment. So our only requirement is all the participants have to attend the assessment. So after attending the assessment, we will be sending the scores to you after 1 or 2 hours of that assessment, so that you can know the correct answers of all the questions. So it is mandatory to attend the assessment on 26th. So please note, and kindly inform to your friends also who are not in the session today. So there will be a session on there will be assessment on 26th. So we are sending the messages regularly to you, like the recordings, the feedbacks, and all other things. We used to send all the things to your WhatsApp, but some participants are regularly saying that they are not receiving. Whenever I ask them, they simply say that they forget to save our number. So as I instructed earlier, participants who saved our number only will receive our messages, because we used to send WhatsApp broadcast message. WhatsApp broadcast message will be delivered to the persons who saved our number in the device only. So I request all the participants to save our institute's number 9488773165. So it is I had sent the number in the chat box also right now. So I request all the participants to save this number to receive our further communications. So also we will be sending the registration number to you by tomorrow evening. So the participant should enter their registration number in the Google Forms, that is assessment form. So assessment form will ask for your registration number. The registration number is nothing but the registration number which we are giving for each and every participant. So we'll be sending the registration number to your email. So kindly check your email. The mail can maybe delivered in some other folders like spam box, promotions, or like other. So I kindly request you all to search your inbox by tomorrow night or day after tomorrow morning using our email ID. Just a minute, I will send our email ID in the chat box. Our email ID is i2tn@gmail.com. So we will be sending the final mail which contains your registration number. So save that registration number so that you can enter in the assessment form. Okay. So these are the two main instructions from us right now. So I request all the participants to attend the final assessment. Just wait. Some participants have joined right now. So I want to tell the brief of today's instructions. So we are having the final assessment on 26th January 2024 from 6 p.m. to 7 p.m. The assessment will be through Google Forms. Assessment form is having 20 questions. In the 20 questions, you have to attend all the questions. There is no passing minimum for that. The only condition is you have to attend all the questions. So participants who attend the test will only, assessment will only get the certificate. The certificate will be issued 3 to 4 days. Soft copy of the certificate will be issued after 3 to 4 days after the completion of the final assessment. So if you complete the assessment on 26th, you will be getting the certificate before 30th January. It will be sent to your WhatsApp, and the hard copy will be sent after 10 days or 1 week after the corrections, all the corrections been made. So this was something we want to say. So with this short note, I would like to hand over the session to Dr. Praveen sir, and I thank all the participants who actually participated in this course. So thank you all. So, sir, thank you, sir. Thank you. Over to you, sir.
Thank you, Professor. So I thank the coordinator, sir, once again for providing me an ample opportunity to conduct this kind of a certificate course on artificial intelligence and machine learning approaches for drug design in cancer. You know, each and every time we are changing the titles according to the necessities for the participants. Okay, like nowadays, the artificial intelligence and machine learning approaches came into existence in so many labs, in national institutes as well as the private institutes, and also we are getting much funding. Okay, so I think I already started presenting. So we are also getting funding for these kind of projects or workshops, FTP seminars, whatever we conduct. So I think Professor Praveen sir, the coordinator sir, has taken appropriate measures to fix a title for this certificate and internship course. Actually, I got one message from a participant that what about the internship, sir? Isn't it? So actually, you know, what is the internship actually? Like whatever skills you gain, okay, you have to utilize it for your project. Or if you already decided to work on a project with your project guide, and I already told you that even I am interested to guide projects. But here, whatever projects I guide in Sri Venkateshwara College of Engineering with whatever infrastructure we have, that is actually charged based on the consultancies. We charge minimal charges as a consultancy charges. We provide to the institution, whatever you pay the fees. Actually, you know, we have excellent facilities for not only for design, drug and vaccine design, we also have facilities for drug and vaccine developments. Okay, so many projects came across, and even we have so many product generated from our institution. We also have an approved CCSEA approved animal facility in our institution. Okay, of course, if you see in Tamil Nadu, this is considered one of the best private institutions to work on research. Okay, and related to this my profile, again I'm showing you this is a profile from an extract from IIT Madras. Okay, I'm working as a postdoctoral DST sub Tare Research Fellow under the guidance of Professor Michael Gomiha, who is India's #1 bioinformatician and considered as top 2% scientist. I think 2 or 1% scientist in the world. Okay, so and about me, okay, about my profile in LinkedIn. Again, I'm sharing you. I think I already Professor has shared you, and this is my profile. And related to my communication email ID. I don't know whether I'll be forgetting to provide all my details, so I'm providing it now itself. So this is my communication email ID. So if you want to work on any kind of a projects, if you want to work on any kind of thing, okay, like you want to work on MTech, MEM Tech in our institution, you want to work for PhD or PDF projects in our institution, you are welcome to work under my supervision. Okay, I've already guiding 3 projects, 3 PhD students, and also the students are receiving stipend from SVC itself, like 18,000 per year, and If they publish a paper, they will be getting 20,000. Okay. And of course, we also, I won't leave like that. I also take you further to write for grants to secure from CSCR, TBT, and also DST. Okay. Definitely, we're also getting a kind of a fellowship meanwhile. So they just register. It takes some time to even generate money from even from the PhD scholars. That is through government organizations. And now let me, before going to take this today's session, that is the last session on artificial intelligence, we can see the schedule, how we have programmed this. So as I mentioned, this is not just a certificate, also an internship can be continued on working with the projects. Okay, and even you can work on your projects whatever your supervisor already proposed. Only thing is that you have to watch the videos and you have to develop the skills. Many PhD scholars, when I visited even a Well-Tech University, some African scholars or completing the PhD with whatever skills they gain from the internship and certificate course. And also I found some PhD scholars are about to submit the PhD thesis with these kind of a computational skills in past Institute of India, Konoor. Okay, likewise, I also know some personal contacts. They're not just limiting to work on UG projects. Also, PG, especially PhD students are getting more benefited from the skills. Okay, so of course they're submitting at least 50% of the computational research, whatever I'm teaching them. Okay, they are focusing either drug or vaccine design, and they are completing these kind of skills, projecting these kind of skills in their PhD thesis. Okay, so I also welcome all the participants, okay, to utilize all the skills, okay, whatever is applicable for your research. Don't just study and just complete the course to merely clear the MCQs. Also, you should have a mind that you can utilize all these skills in your research. Okay, so this is my suggestion to everyone. Now, let me take you to the schedule how we are planned. So initially, if you see the first 2 days, 2 sessions we have discussed about target discovery, and later we have discussed about 5 sessions on drug design. Okay, so if you see the target discovery, we have discussed on introduction to biological databases, target identification, modeling, and validation. Okay, so initially we have seen about the oncology databases. Next, we have seen about protein targets, and if the structures are not known, how to model it and how to validate the 3D structure of proteins. Then we have discussed about methods of computational methods of drug design. Almost 11 to 12 methods we have discussed, and we also seen what are the drug databases and drug likeness and ADMT properties in the 4th session. Then we have discussed about molecular docking using Autodoc. Then we also see now how to go for molecular docking analysis using Discovery Studio, virtual screening methods. We have seen in Autodoc PYRX, and then energy minimization molecular dynamic simulation using GROMAX. And we have seen the basics of cancer biology. And we also seen what is the application of the genomic study and epigenetics study in cancer biology. And then we have in the system biology, in the final session, we have discussed about network pharmacology and how about the how to interact with the pathways, how to see the pathways, and how to link one pathway to with another pathway using the cytoscape visualizer, using the cytoscape network visualizer. And then we have seen in vaccine design about the concepts of reverse vaccinology and basics of immunological databases. And then we also discuss something about T cell epitope vaccine design and B cell epitope vaccine design, especially for Nipah virus, whatever paper we have published. Okay, even for if you see the network pharmacology and pathway analysis, we have discussed with the already papers on diagnosis and treatment of ovarian cancer, and also similarly the diagnosis of first and second stage of kidney cancer. So whatever we discussed from the beginning, okay, so we have I have provided with suitable examples in the recent journal papers published, okay. And here also I've used a Nipah virus kind of vaccine. We have designed it for Nipah virus, and even this project got selected for base Tamil Nadu project, TNSST grant we have received. And not only that, we also this project got selected for INE National Award. Only 2 projects got selected in all over the India. And of course, this is one of the project got selected. And if you see this kind of a machine learning techniques on yesterday, we have discussed something related to MATLAB. You remember, I also told you what are the different online courses available in MATLAB to learn about the machine learning techniques, and how about working on pharmacokinetic model. If you're working on tumor growth model in the mice and whatever results you generate in the in vivo experiments, all these results you can feed in the already developed model. We also have a sample file downloaded from internet. So just like that, you have to feed the input parameters of the in vivo experiments, whatever you have completed related to drug design or vaccine design, and then you have to with a developed training model, whatever values you fit, you get an answer, or you get a dose response curve. That is, you can measure the bioavailability of a drug, okay, even using the MATLAB. There was so many kind of curves, okay, so bioavailability, bioequivalent studies, all these things we can also work on MATLAB. Especially, you remember, I have explained something about SynBiology tool, how to use SynBiology tool in MATLAB. And also, if you see in MATLAB, we also have a special machine learning and artificial intelligence tools. We also have that kind of a toolbox. We have bioinformatics toolbox to work on especially bioinformatics projects, especially on next generation sequencing. Even if you have gene expression data, you can also use the gene expression data to work on the MATLAB. Even the papers, whatever I published in scientific reports and informatics in medicine unlocked and Elsevier journal, I have utilized the MATLAB software. Okay, so this is what we have completed up to the last 13 sessions, and of course the final session is with the whatever sessions we have conducted. Only the MCQs whatever I have developed is from the basics of whatever 14 sessions. I just started from first day to present day. Okay, so only basic questions you can expect. Okay, so even this MCQ is easier to attend. Only thing is that you have to learn the basics from the recorded videos whatever we have shared to you. Any queries before starting today's session? If no, I can take you to the PPT. So today's session is about artificial intelligence approaches of drug design. So I already told you that I'm working in protein bioinformatics lab under the guidance of Professor Michael Tomiha from Department of Biotechnology, IIT Madras. And I already told you even I have developed my own database in C. Venkateshwara College of Engineering. So I utilized, of course, machine learning algorithms, and majorly I've used this kind of languages, C# SQL and PHP. And if you see under the Professor Tomiha sir has developed more than 25 tools, not only in proteomics, also in DNA RNA interactions and RNA drug interactions. So many kind of interactions he worked on with the macromolecules. Okay, so even if you want to develop, if you have an idea to develop any kind of databases related to drug and vaccine design, so you are encouraged to apply either to work on my lab or even IITM protein bioinformatics lab. And related to journal publications, I told you only from the publications I have provided you the skills, hands-on experience, like what are the results and how we got the results. All these things we have started from the day one. So artificial intelligence. Now we can come across the broad chapter called artificial intelligence. So artificial intelligence is of course a larger discipline, and machine learning is one of the branch of artificial intelligence. That's what we have seen on the last session, isn't it? So artificial intelligence is what the main, okay. And here, what is it actually? It is a software program that has been trained on a set of data to perform specific tasks like recognizing certain patterns. So generally, the artificial intelligence in biotech, majorly they are using for diagnosis of diseases. But the people, those who know how to use artificial intelligence in drug design, they are also using well. Okay, so many of the softwares, whatever developed in docking, in pharmacophore design, or in terms of drug likeness prediction, and almost all the softwares, including protein structure prediction and even a peptide structure prediction, even a small molecule structure predictions from the SMILES format. All these things are developed only through the artificial intelligence, isn't it? Just like that, drawing a compound and you'll be getting a SMILES format for that, and you also can get a 3D structure in the software tools. How is it happening on all these things? They have already implemented the AI in the tools, okay, the online tools. So how this can be done? Actually, we have so many models. Even yesterday, we have seen whatever models we use it for machine learning. Of course, we are using the same models for developing this kind of an artificial intelligence tools. So we are using linear regression, deep neural networks, logistic regression, decision trees, linear discriminant analysis, naive Bayes, support vector machines, learning vector, or quantization, and backing and random forest. Each and every part of a drug design can be designing a drug, designing a protein, designing a RNA, whatever you have. Okay, so all these things need a kind of an AI. Okay, especially if you see, if you want to target SARS-CoV-2, spike glycoprotein is one of the important target. But other than the spike glycoprotein, if you see, proteases and polymerases are also the important target proteins. Proteases, we have chymotrypsin like a proteases even in SARS-CoV-2, and also we have DNA dependent. It's an RNA virus, isn't it? So you have RNA dependent RNA polymerase and RNA independent RNA polymerase. So if you see RNA dependent RNA polymerase, RNA molecule is bound with the RNA polymerase protein. Okay, now you are talking just the polymerase, but also the RNA binding regions of the RNA polymerase. So if you want to see the, even if you know the structure of the RNA, then only you can understand even the therapeutic targets. Okay, so for this also you need an artificial intelligence. It's not just limited to develop drugs of small molecules. We can also able to develop mRNA kind of molecules as a therapeutic targets, even using this artificial intelligence. If you see the father of artificial intelligence, John McCarthy, who is an American pioneer computer scientist. Okay, we call him as a father of artificial intelligence. What is his major role? He worked on the field devoted to the development of intelligent machines. Okay, so the and if you see this artificial intelligence, there are so many applications with respect to drug design. So what are the various applications of drug design in artificial intelligence? You can find the applications in drug discovery and various drug design topics, and for this we are using various artificial intelligence models. If you see in drug discovery process, especially for target identification and study, we can use it for prediction of protein-protein interaction networks. Not only that, if you want to predict the protein folding, okay, in protein folding, one should have a knowledge on kinetics, protein kinetics, like however you study the chemical reaction engineering in the form of zeroth order, first order kinetics, second order kinetics, third order kinetics. Similarly, even for the biomolecules, like especially the macromolecules like protein, we also follow all these four order of kinetics. So the protein folding is based on the kinetics, of course, based on the various constants attaining the equilibrium. Also studied about thermodynamics, isn't it? In thermodynamics, we can come to know that a system and surroundings. These are the two important terms. Here, if you see, the protein is your system and surroundings is the kind of a solvent or a buffer where you keep hold of a protein for a long time. To make a protein for a long time, you will be placing it in the buffer, isn't it, in general experiments? So you can come to know about even the protein folding. Not only that, based on the various change of parameters, if you change the pH, if you change the temperature, if you change any other kind of buffering solvents, what is going to happen with respect to protein folding? Okay, all these things we can able to work on simulation, computer simulation. Okay, no need to go for in vitro or in vivo. Simply with the in silico experiments and computer simulation techniques, especially even using the PROMAX program, we can also predict the protein folding. Okay, of course, all these things uses the kind of an artificial intelligence. Even in PROMAX, whatever we are working in workstation in a PB, protein bioinformatics lab in IITM, one should also have a knowledge on Python programming. So knowledge of Python programming is needed, of course, even to work on simulation part. Okay, so of course, if you see the Python, it's more related with the artificial intelligence. Many artificial intelligence programs are designed using Python. Okay, of course, if you want to link with the large amount of data, big data, we are linked with cloud computing, and of course, we link with the TensorFlow. Okay, so if you see from the small scale to the large scale, obviously we we rely on the kind of an artificial intelligence tools. And if you see this kind of AI models, we use FT, DCA can generally detect the druggable sites, and CNN, of course, in the form of neural networks, Okay, conservative neural networks helps in predicting the residue. So the expansions of all these terms, whatever abbreviated, CNN, RNN, DNN, FTDCA, all these things are mentioned in the bottom, the adversarial autoencoders. Now, let me take you to the second. That is, if you want to discover the hits, I mean, what is the potential lead compounds? Okay, the drug molecules used for targeting a specific target protein. So for this, you should have a knowledge on the drug repurposing. And if you have plenty of compounds, you have to screen a base compound on binding analysis. Of course, these are we call as in virtual screening. And even if you have plenty of compounds, but you don't have an appropriate targets, you want to calculate it general, that is based on the ligand alone, the ligand activities alone. Even you can use QACR kind of analysis that is based on activity score. So virtual screening, you should have both a 3D structure of a protein and a ligand. But here in activity scoring, just if you have a 3D structure of ligand, that is sufficient for you to go for activity scoring. So this is something about hit discovery. For these kind of techniques, we can use these kind of artificial intelligence models. For example, for drug repurposing, one should have a knowledge on network pathways and about the network pathology of a specific disease, okay, of a specific cancer in our case. And if you see the virtual screening, for virtual screening, we can use so many kind of AI models, especially support vector machines. Okay, support vector machines, of course, if you want to check your drug molecule is going to bind in the lipophilic region or hydrophilic region. You remember on yesterday, I was explaining you that SVM is implemented in Swiss ADME tool for prediction of ADME properties. Okay, of course, that's a kind of a virtual screening. You can make it out. And similarly, adversarial autoencoders. This is something related to genetic engineering algorithms. Okay, so where you can use this even for virtual screening, especially Autodoc Pyrex is using this kind of algorithms. And if you see the next kind of a drug discovery process, hit to lead. Okay, so we are using the QACR. Actually, the QACR is of course a kind of an activity relationship studies between the different ligand compounds. Just the 3D structure of ligands is sufficient to study about the QACR, like how you go for calculating the activity. Of course, these two are interlinked. And if you see de novo drug design, where you can also design the drug in computational, where you use deep reinforcement learning. That is, if you want to get a kind of a drug, especially if you see de novo drug design is something linked with the earlier stages of drug design, like something linked with the pharmacophore design. Okay, so something related to the pharmacophore drug design, like if you know the functional groups and the activity of the functional groups in a particular backbone or a lead compound, then you can able to identify the best drug. Okay, like if you attach or detach the functional groups in the lead compound, then only you can able to find which is the best drug you can able to get a drug hit you can get for a particular receptor. Okay, so that even de novo drug design helps in identifying the drug by finding the potential finding functional groups in the backbone structure of the lead compound. And nearly and next also lead optimization. This is used for evaluation of ADMT properties. Of course, we also seen the support vector machine is also using this kind of SVC ADME tool. We are using support vector machine to calculate the ADMT properties. But other than SVM, we can also use conservative neural networks and recurrent neural networks and multitask neural networks for calculation. If you see the applications of AI in drug discovery and development, the applications of AI we can see also works in clinical trial design and monitoring. In SRM institutions where you find engineering, pharmacy, and even the medical streams. Okay, we have different branches, isn't it? Like SRM, we have few more colleges where you can find all this. So they are also giving employment for the UG students to work on the clinical trials. Okay, of course, if you want to work on the clinical trial kind of a softwares, that is by taking the subject enrollment of a patient, and after the patient discharge, you also have to enter all the details of the patient before they're getting discharged and monitoring the trials, whatever drugs they're treated. while they're admitted in the hospital. So all this kind of a clinical trial design monitoring, of course, they are using a kind of an AI tool. Okay, we already have a packages. Okay, we have having the clinical trial packages to work on. So these packages, the software packages are developed based on the AI principles. They are using various machine learning techniques to work on entering the data and to give a kind of a result in the form of a graphs. If you want to analyze various graph analysis, they're using plenty of tools. And if you see this kind of pharmaceutical product management, that is, if you want to in the after the phase 3, if you go for phase 4, post marketing surveillance, of course there also you can use the AI for software development. And related to quality analysis, assurance, quality assurance and quality control. Okay, even HCL and other companies are giving much jobs for the UG scholars, from the UG scholars onwards. Okay, so they are also utilizing these kind of principles like understanding critical process parameters, guiding future production cycle, regulation of inline quality, ensure quality assurance with respect to ELN and other techniques. All these things, even the tools, whatever they develop from carried from the AI. And pharmaceutical manufacturing, automated manufacturing, personalized manufacturing, correlating manufacturing errors to set parameters. They are also using the AI. And similarly, drug discovery, whatever we discussed, so related to drug design and related to drug screening. In drug screening, not only by activity, toxicity, and other physicochemical properties, including ADMT properties, are determined using the AI. And if you see the pharmaceutical product development, also we are using AI. Okay, this is an extract taken from the Drug Discovery Today journal. And in artificial intelligence for drug discovery for your market development. So not only we are using in specific applications, we are using in almost all the applications I mentioned. That is for the prognosis, diagnosis, not only used for treatment, also prognosis, diagnosis. We have to use the artificial intelligence. And how we are using the AI. So only from the data, whatever taken from the patients. Generally, if a patient is admitted in the hospital, immediately what they tell the cancer patient, if they are admitted in the hospital, they ask them to take X-ray. They want to take ECG, EEG. So different parameters isn't it? Not only they are going to take the digital data, but also they take an analog data, like whatever they record from EEG, ECG. And obviously, all this analog data has to be converted in the form of a digital data, and even they have to come up with a kind of a conclusion from the software itself. Okay, so this is happened. This can be worked using the AI. And in India, we don't have EMI. Okay, EMI kind of a databases. Like we have a Aadhaar card, we have PAN card. Okay, so where we can get the entire details, including our bank account details from these major identity cards. But in the developed countries, they're not just limiting up to how to extract the kind of income tax from these kind of identity cards, but they also have health identity cards. Okay, so each and every patient, each and every person, citizen have a patient ID cards. With the patient ID cards, if you just you also have a kind of a QR code. If you scan the QR code, obviously we can get the detailed history from the history of illness. Okay, like during any child during the birth, okay, or the womb, in the mother's womb. Okay, what drugs your mother has taken, and even after the birth. Okay, from day 0 or day 1 onwards. Okay, whatever what drugs are taken for what kind of a disease. All these things are recorded in EMA, isn't it? Electronic medical record contents. So all these things can be maintained. That type of patient reports and type of drugs they have taken, how many days they have taken for relieving from the particular disease. All these things can efficiently we can check on the artificial intelligence tools. So all these hospitals in USA works in the form of a network, and all this data are interconnected. So like if a patient is approaching one hospital and they want to change in another hospital for a different disease, even if they go to the different hospital, if they scan the code, they will be getting all these details, the previous illness, etc., and even the drugs, whatever they taken. So there is no the time taken for the clinician to diagnose and to give prescribe a drug is reduced. The time taken will be reduced drastically. Okay, but in our country, we are here to bring this kind of a patient ID cards and the electronic medical records, where all the hospitals has to connect in the network to with the details of the patients, the history of illness, and the drugs, whatever they're chosen for the illness. Okay, so all these things are yet to be developed. We have still plenty of opportunities to develop these kind of EMI, the EMR records of the patient ID card. So we can anticipate these kind of techniques can be used not only to come up with the drugs from the clinicians within a short span, but also we can able to get appropriate diagnostic measures, appropriate prophylactics, and other kind of prognostic measures from the AI. Now, let me take you to the next slide. So this slide tells something about repurposing databases. So repurposing, like generally, if you see the AI, okay, applications in discovering a drug for SARS-CoV-2, okay, that's an example I'm mentioning. So initially they come up with a repurposing tax, isn't it? So of course, for repurposing, you should have a knowledge on AI. But what are the different repurposing databases already publicly available in the net? We have for chemical structure, these are the databases, the general databases for drug molecules, for drug targets, so for protein-protein interactions. Of course, these are the repurposing databases developed based on AI. Excuse me. And in silico approaches in drug repurposing. Remember on yesterday, I was explaining you the in silico approaches like the drug-based. In drug-based, we have chemoinformatics, determining the side effects, metabolomics, drug gene similarity, structural similarity, pharmacophore similarity. So these are one of the essential techniques in drug-based drug repurposing. If it is a disease-based, we should have a knowledge on data mining, text mining, gene signatures, metabolomic data, epigenetic data, network pharmacology, phenotypic data. For each and every data, you may have different web resources. Of course, we should not only use all this, we should also use disease-home networks, okay, where how one disease is connected with another disease. Even this is one of the recent trends they are developing using the AI tools, the disease-home networks. So we have PPA networks, we have drug protein interaction networks, we have gene networks and gene regulation networks. Not only interaction networks, gene interaction networks, we also have gene regulation networks. But now even more studies are going on even to establish the disease-home networks from the pathway networks. Okay, and if you see this target based, okay, we have a network pathway analysis, data mining, molecular docking, and dynamics, and even the gene expression data that is using bioconductor tool, okay, to determine the gene expression from microarray data sets. Okay, and of course even NGS, whatever you work, okay, of course all these things you can find something related to both disease based as well as the target based. And if you see the pipeline of AI-based drug discovery development of COVID-19, of course, if you see this kind of identifying the targets, especially I told you, not only the spike glycoprotein is an important target, but also you find main proteases and RNA-dependent RNA polymerase. So these are the other two proteins, important target for the COVID-19 disease, that is for the SARS-CoV, various SARS-CoV-2 strains. And if you see for this kind of thing, okay, if one has to develop a kind of a drugs, okay, or a vaccines, here for the drugs of small molecules or drugs of antiviral peptides, okay, so one should have a knowledge on this kind of a machine learning methods, okay, linked with AI. And similarly for the vaccines, for developing T cell, B cell epitopes, whatever we discussed on last sessions, okay, we are using deep learning model, traditional classifiers, docking, etc., to come up with a vaccine. So If you see the overall computational drug repurposing methods, okay, we are using, of course, network-based algorithms for the drug target networks. I told you there is one database called Stitch database. In Stitch database, you can find the drugs or interacting with some target proteins. Okay, one or two drugs, or even we have a maximum of 3 drugs interacting with multiple target proteins. Okay, that is a Stitch database. So not only that, the recent developments of drug repurposing came with even with a host interactomes. The interactomes are all developed in the last 12 to 15, 12 years. Okay, the interactome networks. Initially, we came up with the genome databases. Later, proteomes came into existence in computational biology era. And in the protein interaction networks, we're also getting so many interactomes, metabolomes, databases, or data are enhanced periodically. Now we are also getting a kind of the other networks like 1,000 genome projects for almost most of the important species. We already have for human, mice, rats, etc. But even also we have 1,000 genome project for Oryza sativa, which is a rice. Okay. We have plenty of rice, isn't it? 70 varieties of rice is there. They're trying to establish all the 1,000 genome projects for almost all the types of rice. Okay, we also have 1,000 genome project for Solanaceae family, especially for tomato, like that. Okay. So these are some of the recent researches happening on. Okay, and of course, if you want to find for any kind of if want to come up with a drug or a vaccine for any type of a newly coming infectious disease, even they are developing algorithms like SARS-CoV-2. If any new strain is going to come, even they're developing the tools to automatically discover a kind of a drug or vaccine using the AI tools. Okay, a lot of complex algorithms coupled to mixed together to come up with this kind of a large software package. And if you see the expression-based algorithms, okay, I told you not only the cancer, most of the diseases need how much expression is happened in the tissues. So they take various tissues, and from the various tissue samples derived from the microarray data set, they measure the gene expression signatures through the intensity of the colors. Okay, they also take you to the, they also take these signatures to the heat maps or the fold change maps, and then they can come up with the percentage of gene expression uh, for a particular patient. Okay, so all these things can be done. Okay, the expression based algorithms can be, of course, these are important for computational drug repurposing and docking simulation. I told you, even in simulation, we're using Python programming. One should also have a knowledge on the Perl programming. So these programming skills also can give some knowledge on how uh, the tools are developed. And even if you want to develop your own tools, you can also slightly modify your algorithms. Okay, and you can work on this kind of a software. Strategies of drug repurposing. So what are the different strategies? So one should have a knowledge on drug, target, pathway, disease, side effects. Then whatever drug design you're coming across for a particular disease, you are going the right pathway. That's what one can able to mention. So what are the methods and approaches one should have? So if you want to go for drug repurposing for a specific type of cancer, I told you the cancer is formed in almost all the tissues. Okay, if you see in your face, okay, so in the face itself, you have a lip cancer, you have a mouth cancer separately, tongue cancer is different, and even bone cancer is different. Okay, of course, tooth cancer. So in the face itself, for each and every part, I'm telling you, we can able to get these kind of a cancer infections. And obviously, if any kind of a peculiar kind of a rare disease, okay, something related to the cancer, if you get, obviously one should have a knowledge on even on this kind of a drug repurposing, because we don't know what is the exact drug we can use for treating at that particular time. Okay, if any new rare disease come, like one in 100 million patient is going to come up with a rare disease, then obviously one should have a knowledge on this kind of a drug repurposing techniques. So the different methods they use: molecular docking for modeling, chemical similarity, fragment-based screening. All these things are computer-aided drug design methods, isn't it? Biochemical assays, cellular assays, animal models. These are essential for pathway construction, protein-protein interaction networks. Okay, and gene expression, protein expression, metabolites, signaling, omics data mining in terms of metabolomics, biomarker-based assay, phenotypic in terms of bioinformatics and chemoinformatics, side effects related to database. Different data drugs are developed based on these kind of a computational techniques. So the famous drugs like aspirin, metformin. Okay, so one is the pain reliever, and the other is for treating the type 2 diabetes mellitus. Okay, all these things, of course, are developed on the computational based methods. Okay. And if you see AI and machine learning approaches together, AI coupled with machine learning, okay, mixed with the machine learning approaches are used for prediction of repurposed drugs for coronaviruses. I already mentioned the different algorithms they have used is support vector machine, random forest, artificial neural networks, k nearest neighbor, deep neural networks. So each and everything are used for different computer-aided drug design. Okay, there are different methods, isn't it? Each and every method is designed based on the different machine learning algorithms. Example of AI tools in drug discovery. There are different examples of tools are mentioned. So one largest, many people are using the DeepChem. Okay, the DeepChem is available in the GitHub repository. So you have plenty of models already developed using the Python-based system. Okay, only thing is that if you want to check your activity of your drug, then you have to upload your drug as in the form of a test data set, and you can check with the already developed training data sets present in this GitHub repository. So like deep chem, you also have, you want to predict the deep toxicity, like there are even, nowadays many universities are developing tools for determining the toxicity. If you see one of the largest database, like developing the toxicity of 12,000 drugs was developed using the deep tox tool. The deep tox tool is going to determine the toxicity of 12,000 drugs, but we have nearly 2.1 billion drugs deposited in GMT+15 database. If you want to develop even for these kind of drugs, for multiple millions of or billions of drugs, then obviously you yourself can develop a kind of an algorithms. Okay, and you can host in the form of a tool. Okay, whatever you develop, AI tool, you can host in the website. Okay, so this is how they developed all these tools based on a particular application that is predicting toxicity, predicting a kind of an activity or a molecular generation. If you want to get a 3D structure from the SMILES format, or even if you want to generate a SMILES format from the structure, whatever you draw in the Java molecular viewer, all these things, okay, or of course, examples of AI tools, especially chemoinformatics tools. So one such patent I recently published in IPR. So that is related to biomedical imaging system for cancer screening using artificial intelligence. Okay, so I'm of course a third author and also published one more patent, design patent. This is related to wireless liver cancer detector. Of course, I'm the second author. So now I'm just taking you to this kind of a utility patent published in IPR last year. So through the biomedical images, like taking the X-ray images, taking ECG, EEG, so all these things, whatever data you generate from the biomedical instruments, okay, we can use the AI, okay, especially to determine the intensity of colors. From the intensity of colors, we can able to identify the particular infection of a cancer in a patient. So this is possible. So for this, I have taken to the 6 claims. Okay, I've taken the 6 claims for this patent, and related to role of AI in drug discovery. So AI is not just limited to use in drug design for these applications. This can be also used in polypharmacology for this kind of an applications. AI can also used in chemical synthesis for these 4 applications, and AI in drug repurposing we already discussed, and AI in drug screening. This is also already discussed. And this I already shown. An illustrative figure, AI in cancer research. How the AI can be used in cancer research, used in diagnosis, prediction of cancer, that is incidence, recurrence, and cancer cell survivals, and also, I mean, cancer patient survival, okay, and cancer treatment. In cancer treatment, drug discovery and repurposing, AI-assisted surgery, precision-based medicine design, most important. I told you for the vaccine design with respect to T cell epitope and B cell epitope, you remember, I have taken especially in Andhra some kind of Brahmin population, and for a particular Nipah virus W protein, I come up with a best epitopic peptide. Similarly, even for a cancer, if you have a cancer protein like EGFR for a particular, if you are targeting on a particular cancer for a specific Indian population, you can come up with a kind of an epitopic peptide. Okay, so like this, okay, kind of a precision medicine, I mean both drug and vaccine. Okay, and a clinical decision support system and self-programming. So AI is used in these various applications in cancer research and AI in cancer early diagnosis. So we can find in plenty of applications, whatever I explained. Okay, I'm just projecting in the form of pictures. That's it. Okay, for image analysis, one should have a knowledge on digital pathology, lesion identification, CNNs, and radiomics and DL classification and prognosis. So generally, from the images only, the doctor can decide and they can tell what kind of a stage of cancer the patient is facing. Okay, the stages of cancer is just how the clinicians can diagnose and tell only based on this kind of an images. Okay, so these images, if they put in the kind of an AI tools, obviously they can get a kind of a precision is kind of an output. Okay, so based on the percentage of expression and whatever they're getting from the software, the doctor can able to prescribe drugs based on the expression patterns. If they're coming with a third stage, okay, they give up with a high dose and come up with a kind of a heavy dosage drug. And if you and if they are coming up with one or second stage, the clinicians can give a low dosage kind of a drug. Okay, and give that kind of a light drugs. Okay, and similarly, biomarker analysis, whole genome sequencing. One should have an idea on the machine learning algorithms and detection, prediction, stratified for the patient trials, artificial intelligence. And one more illustrative figure, that is biomedical imaging in cancer detection. So I told you, even if you are extracting a serum, and from the serum, if you are taking the cells, and if you are checking in the microscopic examination, okay, even through the microscopic examination, through the intensity of colors, and based on how many cancer cells are available based on the cell count, okay, and even by passing it through the FACS analysis, even from the FACS analysis, fluorescent activated cells order analysis, whatever you analyze the cells, okay, the types of cancer cells, okay, you can analyze it through the FlowJo software, isn't it? If one has worked on flow cytometry, they might have knowledge on FlowJo software, which is used to determine the cell count. Okay, so even this FlowJo is of course a product of AI thing. Okay, we are using a plenty of machine learning algorithms to identify the cells based on the shape, size, and how much diffraction patterns it can show. Okay, so however a flow cytometry principle is applied to identify the cells, the same principles we can also use it in developing the software. An illustrative figure for the diagnosis of lung cancer. So if you want to detect the stages of cancer, of course, you should need the tissues, the lung cancer tissue after the biopsy, or even you can take the blood and serum and you can measure the drug accumulation, how much has happened for the treatment. So all these things we can make it out, okay, using the AI tools. So if you want to measure the gene expression, if you want to measure the bioavailability or bioequivalent studies, one should have a knowledge in AI to develop the tools for analysis. And an illustrative figure for breast cancer. This is from this article. AORL is mentioned. Okay, so for the different patterns, okay, so the color patterns, the one can able to identify the not only identify the stages of cancer, but also the genes responsible for causing the cancer. Okay, if you have thousands of genes, what are the most important genes which causes the cancer? Even this can be detected using the AI tool. An illustrative figure for the head and neck cancer. Okay, so one should have a knowledge on different machine learning algorithms. Okay, so of course, the sources like the causes will be like cigarette smoking and some kind of drinking alcohol. This can create this kind of even head and neck cancer. So generally, what we have to do, we have to go for the biopsy. We also have to go for the serum analysis, and then we have to use the machine learning techniques to identify the patterns. Not only we have to go for image classification, based on the image analysis, one can able to come up with the appropriate diagnosis, and based on the appropriate diagnosis, the treatment can be done. Say for drug repurposing in an integrative context. So all these tools, prognosis, diagnosis, treatment, coming up with all these or integrated one each other, isn't it? So this can be done even through these AI tools. And AI, at last, this is the last slide for our entire internship and certificate course. The last slide, I have mentioned that AI is generally we are using it for precision-based medicine design for cancer. We can't able to use one drug even from the repurposing technique or even from whatever the vaccine design based on the epitopic thing. So that using whatever techniques, whatever computer drug design techniques you have, so the general techniques can't be used for treating all the patients. So each and every patient has a unique set of genes, especially MHC genes. I told you now in vaccine design. So one should get the gene signatures. Okay, and even the gene signatures responsible for causing the cancer. Okay, so if only if we start from the scratch, okay, then only we can come up with a drug design or a drug discovery or vaccine discovery. Okay, so one should have a knowledge on precision-based medicine design for cancer treatment nowadays. Okay, not a common drug can use to treat all the patients for a particular type of a cancer. Okay, so this is something about the large artificial intelligence approaches of drug and vaccine design in cancer. If anyone have any doubts on today's session or the previous sessions, you can ask me, because today is the last session. Where after this session, the last session will be directly the assessment on MCQs. If you have any questions, you are welcome to ask. You can ask anything, not only based on the technical knowledge, whatever you gained or yet to be gained from the recorded videos. You can ask me any questions. I can answer for 2 or 3 participants. If no, thank you very much for the interactions. Okay, from the beginning. So you made me more interesting only through the interaction. So even in the last session, I don't know how much I can able to bring. Okay, I want to deliver everything. So I just without a gap, I just delivered all the information. Okay, if you have any doubts, you can also reach me through the email ID. Okay, whatever I've provided. So all the best for your career enhancement and also the professionalism. Okay, so have a great day. Keep in touch with me if you desire.